My name is Davide Michieletto. I'm a reader in statistical physics and the group leader of the Topologically Active Polymers Lab. Welcome to my exp. DNA topology specifically is the study of the topological properties of DNA. And this is really fascinating. There are three main, let's say, topological objects that are formed by DNA. So the first one that is a little bit more complex to understand is supercoiling. And this is because DNA is a double helix. So because DNA is a double helix, what you can imagine is to tighten the helix a little bit more or release the helix. So it's a right-handed helix in particular. So if you keep rotating right-handed, so it will become supercoiled positively, so over-twisted. You over-twist the helix. But if you rotate, if you imagine to hold one end of the DNA and to rotate in a left-hand fashion, the bottom of the DNA, then you under-twist the DNA. And these two processes, they drive supercoiling. So what it means is that the DNA helix, to compensate for the excess or the under-twist, it will coil onto itself. So you see this sometimes when you have telephone cords or even little pieces of string. So if you start to over-twist them, sometimes they start to coil onto themselves. And this is really also what DNA does inside living cells. In bacteria, DNA is intentionally negatively supercoiled. So what that means is that bacteria like to keep their DNA underwound. And this is because if DNA is underwound, so imagine taking the double-strand helix of DNA and you hold one end and you turn anti-clockwise on the other hand. So what happens is that the two strands, they become slightly more open. Okay, so they are less packed into this helix. The helix is a little bit more relaxed. It's more open because it's a right-handed helix. And so for this reason, what happens is that when you open up this double helix, it's easier for other proteins like RNA polymerase to bind the DNA and read the sequence of DNA. And so by keeping the DNA negatively supercoiled, so by managing its topology, you can facilitate gene expression, for instance. DNA knots are present in many different organisms. You can form DNA knots in the capsid of viruses. So if you ligate the ends of DNA in the virus capsid, you can form knots, very complicated knots. You can form knots also in chromatin, so in eukaryotic, in yeast cells. And DNA knots is sort of a knot is something of this type, right? So it's a curve that entangles with itself, and you cannot really transform this into a simple circle without cutting or gluing something. So by doing continuous deformations, you will never get rid of these crossings here. And the third structure is linking. And linking, I can show you with my hands. So linking is this type of topological structure, and this is present in DNA of parasites, so called the trypanosomes. So these are blood-dwelling parasites. They carry nasty diseases in Africa and also in South America, like Chagas disease or sleeping sickness. And the mitochondrion of these single-cell parasites contain a structure called kinetoplast DNA that is made of thousands of DNA rings that are all interlinked together. We don't know why. We don't know what's the evolutionary benefit of having such a topologically complicated structure. But we call these structures topological structures because you cannot really deform these interlinked rings into separate rings without cutting and gluing. And so this is what renders DNA topology fascinating. So it's present in parasites, bacteria, viruses, higher order organisms. And what my group does is to try and understand how DNA topology impacts DNA function, and also how can we use DNA topology perhaps to make new materials. So what is really important and what essentially every living organism, every bacterium, every higher order organism has is one form of topoisomerase. So topoisomerase is a fantastic protein. The first time I heard of it, I thought it was magic. It's a protein that can bind DNA and transiently create a cut, open DNA, pass another strand, a double strand of DNA through, and then glue it back. So it can actually do topological operations. It can open the DNA, can cut DNA, and can close it back. So it can do those operations that otherwise by doing smooth deformations, you will not be able to remove knots and entanglements, but topoisomerase can by doing this transient break, cutting and gluing. And so the fact that every living organism has a form of topoisomerase tells you a lot about how important it is to manage entanglement and the DNA topology in the cell. It's really essential for life, and when topoisomerase do not work, it's when we get cell death or cancer cells. So in cell division, what happens is that chromosomes are brought together. So sister chromatids are brought together into the famous X shape. They're brought together at the chromocenter, for instance. To get to that state, so to that pre-mitotic state, let's say, before the two chromosomes are divided into the daughter cells, what happens is that the interphase, so that mass, that packed dense solution of chromosomes have to be disentangled. And if it wasn't for this topoisomerase, what happens is that the chromosomes remain entangled also during mitosis, during cell division. And when the two cells start to divide, then they create a lot of tension and DNA breaks. And when the DNA breaks, then what happens is the cell tries to repair them, and sometimes repair them incorrectly, leading to cell death or to cancer. DNA topology has an effect not only on the function of DNA itself, but also on how drugs work and how we can come up with medical applications to target DNA topology to treat a disease. So for instance, we can inhibit what topoisomerase does by coming up with drugs that target topoisomerase. So for instance, a group of drugs called quinolones are very strong antibiotics, and these quinolones are targeting DNA gyrase, so this equivalent of topoisomerase that keeps DNA negatively supercoiled. Another very potent anti-cancer drug is called etoposide, and this was one of the first anti-cancer drug ever developed, actually. And this targets type 2 topoisomerases, which are the ones that resolve these entanglements. So what happens is that when these drugs bind the protein, or certain active sites of the protein stops working. It cannot resolve these DNA entanglements anymore, and these DNA entanglements, when the cell divides, they transform into DNA breaks. And then eventually the cells die. The issue with these drugs is that DNA topoisomerase are everywhere. They are in cancer cells, they are in diseased cells, but also in healthy cells. So what happens is that these drugs target all the cells, all the topoisomerases in both diseased and healthy cells, and the problem is that they have a lot of side effects. And so one of the big challenges in the field of DNA topoisomerase and DNA topology is to try to discover new drugs that target selectively topoisomerases, for instance, the topoisomerase in bacteria, but not the ones in humans, or the ones in cancer cells and not the ones in healthy cells. There are also technological advances where we can use topoisomerases to cut and glue pieces of DNA into genomes. And this is very similar to what also CRISPR does, or recombinant DNA, so using recombination techniques. This is a way to do genome editing that now is particularly popular topic after the discovery of CRISPR. DNA topology is not always a bad thing. It can also lead to enhanced, let's say, functionality.